Hello everyone, myself Komal Jamdar, student of Ashokram Mani Institute of Pharmaceutical Science and Research, Save. This video is guided by Mr. Azad Dhagesar. I am presented topic explained in detail structure of bacteria. Introduction. Bacteria are microscopic unicellular prokaryotic organisms. The study of bacteria is called bacteriology of branch of microbiology. All bacteria are unicellular organisms that reproduce by binary fission. Bacteria are defined as microscopic single-celled organisms that can penetrate into healthy tissue and start multiplying into vast numbers. They can be found in different types of environment. Evolution of bacteria. Bacteria are considered as the first life form to arise on the earth about 4 billion years ago. All other life forms are evolved from the bacteria. The hyperthermophilia of about 2.5 to 3.2 billion years ago was the ancestor of bacteria and archaea that are found in present time. Symbiotic association between different bacteria around 1.6 to 2.0 billion years ago gave rise to the first proto-eukaryotic cell which gradually gives rise to eukaryotes. External structure of bacteria. It includes a cell wall and all structure outside the cell wall. First, flagella. Second, pili and fimbria. Third, capsule. 4. Sheath and prosthesis. 5. Cell wall. First, flagella. Flagella are long hair-like filamentous structure of about 4 to 5 micrometer long and 0 to 0.1 to 0 to 0.3 micrometer in diameter. They confer motility to the bacteria. Flagella are divided into three parts: filament, hook, and the basal body. The filament is the thread-like part extending outside the cell wall. It is made up of flagellin protein. The hook is short curved structure that joins filament with the basal body. It produces repulsion like the propeller during the revolving the flagella. The basal body is a set of rings embedded in the cell wall and plasma membrane. Second, pilia and fimbria. They are the short, hollow, non-helical filamentous structure of about 0.5 to micrometer in length and 0.01 micrometer in diameter. They are exclusively found in gram-negative bacteria. They are composed of protein filling arranged non-helically. They are short, numerous, and straight than flagella. Sex pili are a special kind of pili that take part in bacterial conjunction. They are larger than usual pili, 10 to 20 micrometer in length. They are few in number, just 1 to 4 in number. They are further classified into two types, F pili and L pili. Third, capsule. It is a viscous outermost layer surrounding the cell wall. It is composed of either polysaccharide or polypeptide of both 29% and water 98%. They are present only in some species of bacteria. The capsule is two types: macrocapsule, capsule with a thickness of 0.2 micrometer or more, and microcapsule, capsule with thickness less than 0.2 micrometer. 5. Cell wall. The cell wall is a structure made up of peptidoglycan that surround the plasma membrane as an external coat. It is a 10 to 25 micrometer in thickness. Peptidoglycan is cross-linked polymer of alternately repeating N-acetyl uramic acid and N-acetyl glucosamine polysaccharide subunits. Gram-positive cell wall. The gram-positive cell wall is thick cell wall containing a large amount of peptidoglycan, about 40 to 90% of the cell wall arranged in several layers. This type of cell wall also contains acidic sugar like teichoic acid, teichouronic acid, and neutral sugar are like mannose, arabinose, and rhamnose, and glycosamine as matrix substance. Teichonic acid are made of polyribitol phosphate or polyglycerol phosphate. They are major surface antigens of gram-positive bacteria. There are two types: wall teichoic acid and lipoteichoic acid. Gram-negative cell wall. The gram-negative cell wall is thin cell wall with significantly less amount of peptidoglycan. It is comparatively more complex than the gram-positive cell wall. It contains lipoprotein, lipopolysaccharide, and outer membrane in addition to peptidoglycan. The lipoprotein layer is composed of lipoprotein. It is embedded in the outer membrane and stabilizes the outer membrane. The outer membrane is bilayered structure containing an inner layer resembling the plasma membrane in composition and an outer layer made up of lipopolysaccharide. It is rich in a variety of proteins like porine and outer membrane proteins. Gram-positive cell wall versus gram-negative cell wall. The gram-positive cell wall is thick and gram-negative cell wall is thin. The gram-positive cell wall is higher peptidoglycan content and gram-negative cell wall is lower peptidoglycan content. The gram-positive cell wall is lower lipid content 2 to 5% and gram-negative cell wall is higher lipid content 15 to 20%. The gram-positive cell wall, the main components are peptidoglycan, teichonic acid and teichuronic acid. The gram-negative cell wall is the main component are peptidoglycan, lipoprotein, lipopolysaccharide outer membrane. The gram-positive cell wall is very few amino acid without any aromatic amino acid, and gram-negative cell wall is wide variety of amino acid with different aromatic amino acid. Internal structure of bacteria. In that first, cell membrane, plasma membrane. It is the innermost phospholipid bilayer just beneath the cell wall, enclosing cytoplasm. It is a thin 5 to 10 nanometer semi-permeable. Unlike eukaryotic plasma membrane, they lack sterols and comparatively have a higher proportion of protein in place of sterols. They have sterol-like compounds called haploids. They contain a wide variety of fatty acids like usually saturated and unsaturated type of additionally methyl, hydroxyl or cyclic groups too. The plasma membrane is equipped with several porine proteins for the transport. Second, cytoplasm. It is a colorless colloidal viscous fluid with suspended organic and inorganic solids enclosed within plasma membrane. In that one, ribosomes. Bacterial ribosomes are of 70 years type of quite smaller than eukaryotic 80 years types. They are made up of two subunits, the 50 years and 30 years. Their main role is to synthesize bacterial protein and enzyme. Second, mesosome. They are vesicular or unbranched structure formed by invaginated of the plasma membrane. Third, inclusion body. They are believed to be storage food. They are of two types. In that one, organic inclusion bodies containing glycogen or polyhydroxybutyrate granules, and second, inorganic inclusion bodies containing polyphosphate or sulfur granules. Third, bacterial nucleus. They are called nucleides. Unlike eukaryotic nuclei, they are not enclosed in the nuclear membrane and lack nucleus and nucleoplasm. It is represented by a DNA molecule either in a closed circular form or in colloidal form. Bacterial DNAs are found either in nuclide as chromosomal DNA or outside nuclide as a plasmid. Thank you.